Okay, welcome back. Any questions or comments? This is where I think we stopped with spin traps. Excuse me. For measuring some free radicals through EPR spectroscopy. We are looking at methods for measuring quantitative measurement of reactive species. We looked at fluorescence methods in some detail because we use them in our lab. We just touched upon the other methods through a list and so on. And then we looked at EPR principle in some detail, and some details on EPR, including the use of spin traps, we have looked at. Any questions, comments on that? You'll also have a chance to refresh your memories and so on and so forth later. But any questions, comments now? Okay, if not, we'll move to the last part of this chapter, which is on biomarkers. Yeah. Oh, it depends on the spin trap. It depends on the spin trap. A few hours is very common. At least yeah, you capture the radicals using a spin trap, and the spin trap is what is analyzed using EPR spectroscopy. In other words, the entire solution. But what the EPR will pick up is the free radical in the spin trap, which has been made into a free radical because it has trapped the hydroxyl radical or the specific radical it is meant for. So if you look at, for example, DPTU, DMPO, DPTU something, DMPO, right? We use DPTU. Strange, I used DPTU more than 40 years ago. Okay, DMPO. DPTU is not a spin trap, and something else, some other chemical, dimethyl pyrrolene oxide or something like that. That's DMPO. That is specific for hydroxyl radicals. So you add it at the time that you want to measure the hydroxyl radicals at that instant, and then in a sample, of course. Then it will react specifically with the hydroxyl radicals and produce a, where is that? Produce a species such as this, or a species such as this with superoxide and so on and so forth, let us say. Okay, so this is what the EPR will catch. So the corresponding spectrum for this is something like this. Although it is specific for hydroxyl. Hydroxyl is 1:2:1, right? But if it has been captured by this, the EPR signal will be contributed by that radical, that electron, as well as the environment of that electron, right? Or there are a lot of nuclei around it, electrons around it, and so on. Electrons doesn't matter, nuclei around it, and so on, right? And therefore everything will contribute to it, and the resulting thing will be this. Only thing is that this is typical. Once you see the spectrum, you know it is, you can associate it with the hydroxyl radical or the spin trapped hydroxyl radical. Yeah, different spin traps are for different reactive species. So this is, it doesn't say which radical it is. Okay, DMPO is definitely for hydroxyl. We have used it for hydroxyl. Some specific for superoxide, some specific for this thing, and so on. It could also cross react sometimes. Then you'll have to be a little careful. You'll get a combined effect. But usually we use specific reactive species. That's not a problem. Any other questions? Okay, then the last part of this chapter, biomarkers, which you might already have some idea of, but you need to clarify your thoughts about biomarkers and so on. Some investigators, especially in medicine, are interested in measuring the outcomes of the interactions of reactive species with the cells or the various biomolecules. Okay, this is trying to understand it at the cellular level and trying to figure out at least the extent of damage for diagnostic purposes. The products of the reactions between the reactive species and biomolecules can be considered as fingerprints of the damage if the following criteria are met. There are known criteria. The first one is that they are unique. The signature must be, the fingerprint must be unique. Not multiple things must give rise to the same fingerprint. They should not be generated by the normal biological processes, only by the disease condition, and they should not originate from the diet that is taken in. If these three are met, then they are a good candidate for a biomarker. Okay. Typical biomarkers are damage to DNA, protein or lipid, which are estimated in various ways in the sample. So, it is pretty much fundamental. You look at the damage extent to the nucleic acid, DNA especially, to the protein or to the lipid, and then that is a good biomarker. The lipid peroxidation, I think I cannot move this, right? So, I will move slightly. Not able to read this at all. Okay. I can read that. Not bad. Okay, the lipid peroxidation is done or is measured. The extent is measured by something called a T-bars assay. Very common assay, well known for people in the field, and in the related field. It is well known. Can you close that? Somebody is coming. Okay. It's well known. T-bar stands for thiobarbituric acid reaction species assay. Okay. So as mentioned, highly popular and quite easy to do, and that's the reason everybody uses it. Right? And it is used as a biomarker of lipid damage in many fields, including food preservation. Remember, we talked about flour damage through milling. It causes damage to the lipid. Even that is measured by T-bars. A lot of lipid peroxidation damages are measured by the T-bars assay and so on. Very popular assay. However, the T-bars has significant limitations in reflecting the realities of lipid peroxidation. This is, if you really want to differentiate finely, then be a little careful about T-bars assay. It will give you a rough estimate, what should I say, a bulked up estimate of the damage. But if you want some fine measurements, then be a little careful. And you can look at your textbook for the complications involved in the interpretations of the T-bars assay. It's essentially you measure, rather you react something and then you measure the product. So, okay, you can look at what the T-bars assay is and things like that if you are interested, just to give you an idea of the biomarkers here. And one of the desired features for a biomarker is that it is easily available, it is easily accessible, or its availability in easily accessible sources such as urine and other bodily fluids, so that the measurement becomes simple. Right? Researchers have used urate oxidation products such as allantoin as a marker of damage. More the damage, more the allantoin levels in urine or wound dialysis, wound fluid or microdialysis fluid, and so on and so forth. This is for urate oxidation. The extent of damage is measured through measuring allantoin, a molecule called allantoin. So, allantoin is a good biomarker or a known biomarker. Good example. Okay. So, that gives you some idea of biomarkers. There are many biomarkers, of course. This gives you some biomarkers of relevance to reactive species damage and things like that in a medical context. Biomarkers are indicators of damage that has already happened. Okay, I am re-emphasizing something that I did in the first chapter, that I mentioned in the first chapter. You should note that the biomarkers are indicators of damage that has already happened. They are certainly good for diagnostic purposes. So much of damage has happened, fine. Okay, but not good for therapeutic purposes, because the damage is already done, and since it is reactive species damage, the damage is mostly irreversible. So if you have a huge amount of damage and you're trying to, you need to interpret the damage appropriately and then decide what to do. You cannot relate 1:1 and so on and so forth. Okay, minimum damage, so minimum this thing and so on, so on and so forth. It's a roundabout way of going about it. You need to be careful about it. It's a direct measure of the damage that's already done, most likely irreversible. Okay, we'll finish up with the last video on this chapter. This is, I mean, rather, before we get into the video, any questions on biomarkers and so on? Essentially, the last aspect of it, you need to understand, not many people do, and be careful about that. Any questions? I asked the others, people who came in a little later, any questions on measurements, principles, and things like that? Details, of course, you can. I don't expect you to remember the details. You can always go and pick them up when needed. Some of you will remember, which is very good, very nice. But the other details, others can pick up the details when it is necessary. You'll have a chance to refresh your memories sometime later during the course also on the measurement methodologies. So we looked at fluorescence in some detail because we use them in the lab. We looked at other methods just in passing. There are many methods, many quantitative methods just in passing. And then we looked at EPR principles in some detail and finished up with biomarkers. So the first 3 chapters are pretty much done. I'm going to ask you to watch a reasonably long video. I think it's about 8-9 minutes long. Good to watch it here, which is there on your, which is the last video. It's a YouTube video on the 3rd chapter, the last video, the 3rd chapter. Summary. Okay. And this is a summary of the first of the information that we have covered so far. It's a very nice summary, well done by Osmosis. Earlier, I think we needed I needed an account on Osmosis to watch it. Then I found a YouTube source, so put it up. Please watch it. I'll also run it once and then continue with the next chapter after you finish. It gives you an overall idea of reactive species, antioxidants, and so on. Nicely done. We're going to start looking at reactive species and cell signaling. It is also known as redox regulation. By the way, redox is kind of a soft term. Okay, it's like saying metabolic aspects. Everything is metabolic aspects, right? So redox is something like that. It refers to reactive species. It refers to energy, something that has an energy angle in the cell, the NADH and so on. So NADH and NADPH, that balance and things like that. The redox ratio is the ratio of NADH to NAD plus NADH, or NADPH to NADP plus NADPH, and so on and so forth. So it's kind of a soft term. Okay, metabolic status. Yeah, that was the term that is not metabolism. Metabolic status is kind of a soft term that used to be there. Similarly, you have redox status. So don't try to pin it down to something. It means many things. Okay. So far, we looked only at mostly at deleterious effects on cells because that is the popular view, as I mentioned in the first chapter. Now we'll start looking at the beneficial aspects. And in fact, the rest of the course is going to be on beneficial aspects. Reactive species play important physiological roles in the cell, which we really haven't touched much. And we are going to focus on some fundamental molecular aspects of reactive species on proteins in the context of cell signaling, especially the proteins with iron sulfur clusters, right? The ones that can interact with the reactive species. In this case, iron can be oxidized or reduced by reactive species. The ones with the thiol clusters, TSH groups, thiol group, for example, cysteine, they can be oxidized or reduced either singly or in pairs. You can have a disulfide bond formation also, and that molecule also can be oxidized and reduced. Depending on whether it's oxidized or reduced, it will work differently, and therefore that forms the basis for signaling. histidine groups, they can be oxidized to form oxohistidine. And I think, yeah, oxidation and reduction, as mentioned above, of the proteins could influence their function either directly. An example is P53, which we'll probably see. Okay. Or it could influence by through the transcription factor. Okay, transcription factor, and thus the transcription of relevant proteins could be affected. So the route is through the transcription factor. The effect route is through the transcription factor. So either the direct effect on the protein or through the transcription factor. Okay. So these three we are going to focus on in this course. This is what is known a lot about. This is not an exhaustive list. You could find others also, although rare. So I mean, I wouldn't rule out other possibilities. I haven't seen much work on the other possibilities so far, but that can come up. Any questions so far? So you have a basis by which reactive species can affect cell signaling by modifying the proteins that are associated with cell signaling. Very classic examples are SOXR, OxyR, and so on and so forth. Just remember these terms. You would have heard them, you may have heard them, but these are very common in the bacterial gene situation. So we'll look at some of these. Any questions, comments? So remember, iron sulfur clusters, thiol groups, and histidine groups. These are the ones that we are going to look at. The molecular level action of either oxidation or reduction by reactive species of let us say the iron sulfur clusters, thiol or histidine has an effect at the cell level or at the pathway level. And we are going to look at some examples, first from bacteria, prokaryote, and then a few from mammalian cells. So that will give you an idea as to some details of the mechanism. And that's why it's going to be a short chapter, and that's more of an introduction. Whenever there's a need, you can go and pick this up. More to tell you that they can play very important roles in cellular processes, so as important as cell signaling. I'll just do one today. I think that will, we're running out of time also, but that will also be heavy, so we'll have to do it slowly. So obvious one, rather the most common one, oxidation of iron sulfur centers. We are going to look at oxidation of iron sulfur centers of the SOXR protein. The SOXR protein happens to be a transcription factor, and we can look at the oxidation by superoxide to begin with. Have you heard of the SOX-RS regulon somewhere? No, no. Yeah, I guess it's too specific. Yeah, we are very comfortable with it because we have worked with it and so on, but I guess it's not so common. So the oxidation activates the transcription of something called a SOX-S gene. The SOX-R gene has the iron sulfur center which can detect the reactive species, and oxidation of that or the reduction of that, oxidation of that activates the transcription of the SOX-S gene to produce a SOX-S protein. And SOX-S protein activates the production of about 100 different proteins that respond to increased reactive species, especially the superoxide. And for example, some SOD, superoxide dismutases, DNA repair enzymes such as endonuclease 4, and so on and so forth. A wide variety of things are activated by SOX-S, are under the control of SOX-S.
So RS here is
Reactive species.
Mitigate the effects of other destructive.
Yeah, no, there has to be a certain level of RS in the cell for it to function properly. And this is one of the reasons why. That's how we are bringing it. See, so far we looked at effect of high levels, predominantly high levels, mostly damaging, right? But you come down a little further, it's absolutely essential for the cell to have it. Otherwise, the cell will not function. That is the point that is being made. And that's a figure from a classic paper in one of these Nature Cell or something like that, Diotro and Toledano, 2007. It's a nice review paper. If you, I would recommend that you read it, but we are going to cover most of it here, so I won't make it required reading. But if you get the chance, take a look at it. It's a nice paper. It's a dense, I mean, not very easy to read, but it has a lot of good information. So this gives you the details. When the SOX-R protein, okay, fine. When the SOX-R protein is attached to, not there, is it? Okay, no stylus tray, fine. Is attached to the DNA, it is inactive, right? And when it gets, this has an ion, oh, this is showing up. This is an iron sulfur center, F2Fe2S. This is just a plus, okay. So it gets oxidized by the superoxide to give you FeS2 plus. So when it gets oxidized, then it becomes transcriptionally active. So that's how this works, as simple as that. Not clear? Not clear. Okay. So you have a SOX-R protein with in a reduced state, ion FeS to Fe2S plus. There's a single plus there. Okay. So in this state, it is inactive, and it won't cause any transcription of the DNA. Whereas when it gets oxidized, it becomes 2Fe2S2 plus, and in this state the conformation changes. If the conformation changes, it attaches to the DNA appropriately and causes transcription. Okay, so that's how the superoxide kind of has a handle on SOX-R, or in other words, SOX-R senses the superoxide, gets oxidized, and becomes transcriptionally active. And the reverse, the oxidized SOX-R is reduced by NADPS through a series of electron transfers. Just one composite step is shown here. Okay, series of electron transfers to give you back the reduced form. So that's the cycle that is set up for the process to occur cyclically. So this is an example by which the reactive species can cause transcription of SOX-R relevant proteins. One of which is SOX-S. We'll look at that later. Okay, which is an important one is SOX-S. We'll look at that later. Any quick questions on this? We'll stop here. We are out of time anyway. We are a minute beyond time. That's a little slow. We'll meet again on Thursday and take this forward through examples. First in the bacteria, I think there are 3 more examples in bacteria, and then a few examples in the mammalian cells. That's it.